Can we predict whether a new drug will throw the heart into chaos without any animal testing, without giving it to a single person, purely inside a computer? Yes. Take every channel we've covered this season, assemble them into one mathematical model, synthesize a full heartbeat inside a computer and compute a single score, high, intermediate, or low risk at a glance. This is CiPA's third pillar, in silico, the virtual heart. First, why we need it. As we've seen, watching hERG alone wrongly condemns a whole batch of safe drugs. The real risk lies in the net effect across many channels. But here's the catch. Patch clamp hands you a pile of numbers. This channel blocked 30%, that one 10. So how do you fold them into a single answer? Is this drug dangerous or not? Human judgment is too subjective. The answer is to hand it to a mathematical model. The classic model is the O'Hara-Ruddy human ventricular model. Scientists wrote every ion channel in a heart muscle cell as a set of equations. How sodium enters, how calcium rushes in, how potassium leaves, how the resting state is held. Feed it the current of each channel, and like assembling building blocks, it synthesizes a living action potential inside the computer, one virtual heartbeat. So how do you feed in the drug? Again, with patch clamp. You measure how strongly the drug inhibits each channel. It's IC50, then, using the drug's blood concentration, usually 1 to 4 times the peak Cmax. You convert that into how much each channel is blocked and pour it all into the model. The model instantly tells you what the action potential of this virtual heart will look like once the drug is on board. The key step is scoring. From the drug-treated action potential, the model computes a metric called Q-net, essentially the integral of the net current across one beat, the net charge. The logic is simple. The lower the net charge, the more the repolarizing force has been eroded, and the easier it is to fall into chaos. Compare Q-net against a threshold, and you sort the drug into high, intermediate, or low risk. The conclusion you can write into a report. But there's a subtlety hidden here. IC50 alone isn't enough. Some drugs block hERG with equal force yet carry wildly different risk, and the difference is binding kinetics. Dofetilide, once it slips into the channel, stays put, trapped tight. Cisapride slips in but comes off easily. Same IC50, yet the former is high risk and the latter much lower. So the more advanced models are fed not just a single IC50, but the full kinetics of how the drug binds and unbinds. That step, too, relies on the fine recordings of patch clamp. Put it all together and you see the value of in silico. Fewer animals, over 20-fold faster, mechanistically transparent, and every step explainable to regulators. It's already written into the ICHE14 and S7B questions and answers. It's the brain of CiPA's 3 pillars, the in vitro multi-channel panel, the in silico virtual heart and live stem cell testing, the part that turns data into a judgment. And with that, this season has traveled from a single tiny ion channel all the way to a whole heart beating inside a computer. Next time, we land on the real star of that third pillar, heart muscle cells built from human stem cells that beat on their own. Follow Channel Observatory, and we'll take them one channel at a time.